Protein synthesis, or how DNA and RNA make proteins. Remember, synthesis means to make or to build. Questions? How does structure of DNA relate to protein synthesis? Where does protein synthesis happen? What exactly is protein synthesis? And what if something goes wrong? First, it's important that you understand there's three types of RNA that are involved in protein synthesis. You have mRNA, which is messenger RNA, rRNA, which is ribosomal RNA, and tRNA, which is transfer RNA. Okay, so we have three different types of RNA that are involved in protein synthesis. Over here we have tRNA, or transfer RNA. Here we're going to have rRNA, or what's known as ribosomal RNA. This is representing a ribosome, which has a large subunit to it and a small subunit to it. The large subunit has an A site, a P site, and an E site to it. And then we have our mRNA, or messenger RNA. Messenger RNA transcribes a complement copy to an original DNA strain. There's two basic steps to protein synthesis. The first step is transcription. This is where you take the DNA strand and change or transcribe its code into a messenger RNA strand. Next is translation. This is where you read the messenger RNA strand 3 bases at a time to see which amino acid is needed. How is it done? Transcription. First, DNA is unzipped in the nucleus. This is done by the enzyme helicase. Then RNA polymerase comes in, binds to the DNA strand, and messenger RNA is made. There are sections of DNA that are not coded for during this process, and those are called introns. The sections that are coded for are called exons. The 3-base code we talk about is called a codon. For example, AUG is a codon, CGC is a codon, and UUU is a codon. The complement or opposite code is called an anticodon, such as UAC is the anticodon for AUG, GCG is the anticodon for CGC, and AAA is the anticodon for UUU. Okay, the second is translation. Within translation, we have 3 different parts. We have initiation, elongation, and termination. First of all, the messenger RNA is read by the ribosomal RNA, and it's read 3 bases at a time, or 1 codon at a time. When a start codon is read, such as AUG, this is where initiation takes place and translation begins. Then tRNA, or transfer RNA, makes an anticodon, or opposite, to the 3-base codon that was on messenger RNA. Then tRNA is going to go and transport the amino acid that the mRNA codon represented, get it from the cytosol, and take it to the ribosome. So it translates the codon into an amino acid. The amino acids are then linked, which is the elongation process, until a codon that means stop is read. That would be the termination. The linked amino acids have now made a polypeptide. Remember, poly means many, in other words, many peptides. So once termination has taken place, the peptide is released into the cytosol to be a protein. Now, we talked about tRNA going out into the cytosol to get the amino acid that the mRNA was coding for. So it reads 3 bases, or a codon, and then it's going to translate that into an amino acid. For example, if you have AUG and you look at the codon chart, you have a start codon. If you have CGU, you look at the chart, you have arginine. And then UGA codes for a stop codon. Here's a picture that is showing protein synthesis. It looks a little bit complicated, but you can take yourself step by step, A through E. Pay attention to the fact that the messenger RNA is being read by the ribosome, or ribosomal RNA, and that the transfer RNA is getting anticodons going out into the cytosol, picking up the proper amino acids, and bringing them back to be linked as polypeptides. Messenger RNA transcribes a complement copy to an original DNA strain. So the ribosomal RNA on the ribosome is going to read that messenger RNA transcribed complement copy. As it reads it, tRNA comes in and figures out what anticodon it needs to go get the amino acid that the messenger RNA is coding for. An anticodon is the same thing as saying a complement, or the opposite. So if it's reading this AUG, then it would have a UAC complement. It would then go and get the amino acid that AUG codes for, bring it back to the ribosome, and then it would move down to the next section. So then the next three codons are read, tRNA comes in, creates the correct anticodon, goes out and gets the correct amino acid, reads. As this is happening, those amino acids that the tRNA is bringing back are being linked within that ribosome. Now, when it's read all the way down to a stop codon, that tells the ribosome stop reading, you can let the connected amino acids free. Those amino acids then are released into the cytoplasm of the cell, and they conform to a specific shape which determines its function. It's going to use its shape to determine what function it's going to be, or what type of protein it's going to be. So the function of a protein is determined by, first of all, which amino acids are coded for, so which amino acids the tRNA brought in, the order or sequence of those amino acids, and then the folding or the shape of that protein. Now, what happens if something goes wrong? Well, you're going to get a mutation. A mutation is a permanent change in a cell's DNA, and there's three basic types of mutations. We have point mutations, and this is where just one base is substituted by another. So maybe there was supposed to be an A, and instead there was a G. An insertion, this is when one or more bases are added that aren't supposed to be in there. And then the opposite of that is a deletion. This is when one or more bases are removed from the codon sequence. So here's a chart showing some examples of different mutations and what they would do. If you look in the middle there at the analogy sequence, it should say the big fat cat ate the wet rat. Well, if you have one single substitution, such as a point mutation, that can be called a missense substitution. And you can see there that instead of saying big, it says biz. Now you have nonsense substitutions, and as you can see, it just says the big rat. So it's missing the middle section of the sentence there. Now a deletion and insertion can cause what we call frame shift mutations. So if you look at the deletion, the word the no longer exists. The E has been deleted, so everything was shifted over to the left, which makes the entire sentence not make sense. So if we were looking at a protein, that protein would no longer be built correctly. And an insertion causes a frame shift to the opposite direction. So there it's not making sense after the word big. And again, this is going to affect what protein is built, or if it can even function. Then we have duplications. If you can see, it says the big fat cat. So we have a duplication of fat. Then we have expanding mutations, and you'll see as the generations continue, that sequence is repeated multiple times. Take a look at the associated diseases with the different types of mutations. It's interesting to see how those are actually in fact genetic diseases. mutations affect the folding and stability of proteins. Substitutions, as you saw on the chart, can cause genetic disorders. And mutations can cause a protein to fold differently, so that changes its shape. It also changes how stable the protein is, and therefore it's going to change its ability to function properly. Some different causes. There's a spontaneous mutation, which is just no known reason. DNA damage, which is caused by chemicals and radiation. And you have highly mutagenic, which is energy such as radiation from X-rays, gamma rays, and yes, tanning beds. Okay, can you inherit mutations? Well, mutations that are within body cells or what are known as somatic cells, they cannot be passed on. But mutations that take place within sex cells, they're going to be passed on, and they're going to be found in every cell of the offspring.